Hereby, I open this academic ceremony in which Vishnu Babu will defend the academic thesis Identification of Metabolic Dysregulation and Transcriptional Networks in Retinoblastoma Reveals Novel Therapeutic Targets, Functional Insights and Novel Therapeutic Strategies Using a Correlative Multi-omics Approach, a long title. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you, dear Prorector, and thank you, members of the Corona. Thank you, my family and my friends who are watching it online. For the next 15 minutes, I'm going to give a brief summary of my PhD thesis entitled Identification of Metabolic Dysregulation and Transcriptional Networks in Retinoblastoma, which reveals novel therapeutic targets. So the thesis study looks into functional insights and novel therapeutic strategies using a multi-omics correlative approach. To begin with, I'll give a brief introduction on retinoblastoma. Retinoblastoma is the most common intraocular malignancy in children, which affects in single eye and both the eyes. Globally, the incidence of retinoblastoma is constant at 1:15,000 to 18,000 live births. In US and in Europe, over the last decade, this has increased in the incidence rate. However, India accounts for the highest global burden of retinoblastoma cases, with 1,500 cases being reported every year. The major cause of retinoblastoma is inactivation of the retinoblastoma gene, which is the RB1, which initiates a tumor development in the retina. As you can see the schematics, this is the retinoblastoma tumor schematics where the tumor fills in 2/3 of the globe. The international intraocular retinoblastoma classification systems retinoblastoma groups to 5 groups, namely from group A to group E, with group A having tumors less than 3 millimeters in size, group B with tumors greater than 3 millimeter size but away from the retina, and then there are group C tumors which are more than 3 millimeters in size, which grows throughout the retina and also has vitreous seeding. They poses moderate risk, but in group D tumors, these grow larger and also causes vitreous seeding, which are small tumors which breaks into the vitreous, and they also causes retinal detachment. And then comes the group E, which is the highly advanced retinoblastoma tumors, which are invasive to the choroid, and they also metastasize to the optic nerve, and they also touches the lens. The group E tumors causes threat to vision and life. The various treatment types of retinoblastoma ranges from radiation therapy, thermotherapy, cryotherapy, intravenous chemotherapy, brachytherapy, surgery, and targeted therapy. Like I said, the group A and group C, the globe salvage could be more than 90%. But in group D, the globe salvage would be around 47%. But group E requires a high-dose chemotherapy to manage the tumors. And if they're refractory to chemotherapy treatment, they undergo the enucleation or the surgical removal of the eye. So the unmet clinical need here is major hurdles the clinic faces are most of the retinoblastomas are advanced at the diagnostic stages, and they also poses variable chemotherapeutic responses. These chemotherapeutic drugs also causes bystander toxicity to the surrounding retinal cells. So we tend to understand that the unmet clinical needs of retinoblastoma involves a biomarker which can predict the disease severity, and also a treatment strategy that is devoid of systemic toxicity to retina. In combination of these unmet clinical needs, we designed a study where the first thesis aim was to understand or identify the biomarkers from transcriptomics and metabolic profiles of retinoblastoma using an integrated multi-omics approach. So what is a multi-omics? So single omics gives us information from single one set biological entities, like an RNA gives an information, or a DNA gives an information using, or a protein gives an information, but this does not complete the entire picture. So multi-omics is an integrative platform where it helps us to identify informations across different biological entities, and it also helps us to identify novel associations within the different omics. So with this concept, we developed an experimental design where we subjected the retinoblastoma tumors for transcriptomic analysis, and the vitreous humor was subjected for metabolomic analysis. And in the transcriptomics, we subjected the total RNA, where the microarray was done for non-coding microRNAs, and for the mRNA expression, we looked at the mRNA microarray, and the metabolomics was subjected for mass spectrometry. In using multi-omics, we further looked at the association between different omic layers, where we looked at the association of microRNA with mRNA and the mRNA with the metabolite. Using this concept, the chapter 2 of the thesis identified a key novel association in the retinoblastoma tumors, where combining mRNA microarray and mass spectrometry, we were able to see the mRNA microarray identified significantly regulated genes in retinoblastoma tumors, where E2F2 was one gene which was upregulated, and hexokinase was one gene which was severely downregulated. And in the mass spectrometry, we were able to find the pyruvate metabolite to be significantly downregulated in retinoblastoma vitreous humor compared to pediatric controls. So here what we see is, using a multi-omics integrative biology approach, we find that hexokinase gene and pyruvate metabolite being downregulated, and these two are key components of the glycolysis metabolism. So this is quite unique in a cancer like retinoblastoma, where the glycolysis metabolism is significantly down. And the microarray results also shows us that the retinoblastoma cancers are significantly high in fatty acid metabolism genes. So this helps us to understand that this cancer has a different metabolic landscape compared to other cancer types. And using in vitro models, the chapter also identified a novel regulation where ectopic expression of RB was able to induce hexokinase expression in retinoblastoma cells, which is independent of its classical non-cell cycle regulation of E2F2. To further unravel the RB and hexokinase interaction, we looked at in the chapter 2, we further, using biochemical assays and in vitro models, we try to understand how this signaling regulation happens. So we further unravel that RB regulates hexokinase through HIF1 alpha, and this induces hexokinase 1, and induction of hexokinase further increases the protein levels of LKB1, and this increase in protein levels of LKB1 further activates AMPK alpha. So AMPK alpha is a metabolic sensor, and upon activation of AMPK alpha, it inhibits the ACC, or the acetyl carboxylase CoA gene, which is a key gene in fatty acid metabolism. So inhibition of fatty acid metabolism further reveals us information using metabolic flux analysis, like AMPK, when inactivated, the retinoblastoma cells have high energy turnover or ATP turnover, but when you activate AMPK using hexokinases or RB, this cell energy is significantly low. So using this as a therapeutic target, we used pharmacological activators of AMPK, which is the ICAR, in combination with low-dose chemotherapy. We were able to see a reduced tumor burden in rabbit models of retinoblastoma, and which also shows that this treatment regime shows no bystander toxicity to the retina. To further look into how these cells program the RB and HK1 in retinoblastoma cells to meet their energy requirements, these retinoblastoma cells, how they use the alternative fuels. So like the figures here shows, alternative fuels are required to meet the increasing energy demands for a long run in an automobile, as we see in our daily lifetime. Similarly, cancer cells uses metabolic fuels like glucose, glutamine, or fatty acids, and then reprogram their metabolism for their survival and growth. Here we see that in retinoblastoma cells, using metabolic flux analysis, in the presence of RB and HK1, these cells have a higher tendency to use glucose as a fuel, but in the absence of RB and HK1, these cells have a tendency to use fatty acid metabolism. Thus, this chapter concludes that in the presence of RB and HK1, these retinoblastoma cells are more favorable for glycolysis, while in the absence of RB and HK1, they utilize more of fatty acid metabolism. The chapter 1, 2, and 3 are interconnected, and they help us understand the altered metabolic phenotype of retinoblastoma tumors and how the novel energy sensing signaling circuit can be used to target the retinoblastoma tumors. In the chapter 5, we further try to look into the advanced stages of retinoblastoma tumors targeting the epithelial to mesenchymal transitional program using microRNA. So like the schematic here, the primary tumor is non-motile and polarized, but they can undergo an EMT translation to become mobile, and then it loses its integrity and adhesion, and then further it helps in metastasis and invasion. So we speculate that or hypothesize that retinoblastoma tumors, the advanced retinoblastoma tumors through the optic nerve, they undergo a EMT program translation, and they get translated to a mesenchymal phenotype. And in this process, they also acquire a chemoresistance, which makes them refractory to the conventional chemotherapeutic treatments. And the mesenchymal chemoresistant retinoblastoma cells further more invasive to the distant organs, which are the brain and bone marrow. In the brain, they form the neuroectodermal tumors, and in the bone marrow, they form the osteosarcomas. So to further understand the EMT transitional program, using multi-omics, again, we profile the microRNA microarray and mRNA microarray. And using multi-omics, we try to understand the association between microRNA and the gene expression. And the key finding of this chapter is we find microRNA 181A5P to be significantly downregulated in advanced retinoblastoma tumors. But at the same time, we see significant high expression of EMT transcription factors. The chapter also identified the association of microRNA 185P to inhibit the expression of the EMT transcription factors in drug-resistant retinoblastoma lines. And we further show that this inhibition reduces the invasion and migration of drug-resistant retinoblastoma cells, and it also improves the chemosensitivity of these cells. And in chapter 6, we further looked at to understand how novel adjuvants can be used to target retinoblastoma tumors apart from chemotherapy. So here in the clinics, they use high-dose topotecan for advanced cancers, which targets both retinoblastoma and retinal pigment epithelial cells, causing a bystander toxicity. So we have a class of small molecule peptides where when we do a monotherapy in retinoblastoma cells, we find that they selectively target retinoblastoma cells based on their charges, and they spare retinal pigment cells. So then we developed a combination therapy approach where we combined the peptides with low-dose chemotherapy, and we found that it was effective in targeting retinoblastoma cells and sparing normal cells. So this we further looked into how these peptides were able to be used in the clinic, and then we find that the peptides were able to potentiate the effects of low-dose topotecan, and thus it causes a synergistic effect. So to conclude, the chapter 2 of the study identified dysregulated metabolic pathways and highlighted the metabolic vulnerability in RB tumors. The chapter 3 of the studies uncovers novel transcriptional regulation driven by HK1 and AMPK. The chapter 4 highlights RB or HK1 as critical regulators of bioenergetic profiles. And the chapter 5 of the study identifies microRNA 185 as a novel intriguing target which can be used for targeting metastatic retinoblastoma tumors. And in chapter 5, we highlight the cationic peptides as a potential therapy to enhance chemosensitivity of low-dose chemo drugs in combination therapy to control RB tumors. Thank you everyone for listening, and I give my word to the prorector.
Thank you very much for your presentation.
Yeah.
It's now time to start with the opposition.
Yeah.
And the opposition will be opened by Professor Webers, who was also the chair of the assessment committee. Professor Webers is professor of ophthalmology at this university.
Dear Mr. Candidate, first I would like to congratulate you and your family and your supervising team with your thesis. You've proven to be a true and rightful member of the scientific community, although you still have to defend your thesis today, of course. And moreover, you are a representative of the young and bright Indian generation that makes your country so proud. Thank you for that. As a clinician, some of your studies are not so easy for me to understand.
Okay.
But you have tried to bridge the gap between the lab and the practice in the discussions in your work. And although you are not a clinician, my questions will be a bit in that direction, leaving enough room for the opposition members to go more in depth on the biological issues. So my first question is about the materials you've used in your studies. I think we have to be grateful to the parents who gave permission to use the inoculated eyes of their children.
Yeah.
The various tables in your thesis show that you only seriously affected eyes from retinoblastoma stage D and E used because stages A to C are probably spared as much as possible. And my question is whether the fact that you used only stage D and E eyes might have influenced the results. Or in other words, do your biological phenomena that you found apply also to the earlier stages of retinoblastoma? Does the tumor change during the course of growth? That would be my first question.
Highlighted esteemed opponent. Thank you for your kind words and the question. In retinoblastoma, as I mentioned, the stages. So we are only able to acquire group D and group E stages because the primary clinical goal is to preserve life and the globe. So we were not able to get into a group A or a B or a C retinoblastoma tumors for our experiments or analysis. But as you point out, it's a valid point that early stages would have a different phenomena of metabolism compared to the advanced stages, but right now we are not sure of it because we don't have samples to analyze it.
Are there any similar diseases that you know of tumors that can be studied in earlier stages that resemble retinoblastoma in a certain way? What do you know of that?
Not of ocular tumors, but yes.
Breast tumor, for instance,
and lung tumors, yes. The early stages has a different, the initiation of the tumor has a different metabolism compared to the once they grow out and once they become more advanced, they slow down. When they don't need to proliferate, they slow down and they don't use the highly ATP production pathways. They just slow down and they happily sit there. So they don't want high energy pathways to feed them. Mm-hmm. So that's one possibility why advanced tumors does not use highly proliferating energy pathways.
Okay. My second question is about the reproducibility of the data. I have two questions. First of all, you use the same eyes in a number of studies, and I can imagine why you did. You want to have an integrated omics picture of the tumors. But are then the findings reproducible, since you had only, I think, like 10 eyes or 15 eyes?
We used 9 eyes and 2 controls for our discovery.
And 3 controls,
yes. So for a discovery cohort, we have used 9 retinoblastoma enucleated eyes and 2 pediatric controls. So the entire mRNA microarray and microRNA microarray are all done at the same time from the same RNA. So then we did the analysis separately. Mm-hmm. So the microRNA microarray was used for a different study to find the advanced tumors and EMT pathways, rather than mRNA was used more for the metabolic, and even for the EMT pathways.
And do you know of any duplicate of your studies in any lab in the world, India or wherever?
Not much. When we started the study, we were the first one to compare it to a pediatric retina, because that was quite rare to get. Compared to most of the studies compares the tumors to the adult retina, because that's the most available source of samples.
Mm-hmm.
But the uniqueness of our study is we compared it to the age-matched pediatric controls.
And about reproducibility, do you think the same applies to a Caucasian type of eye? Do you expect differences?
Not sure. Once we do the analysis, yes, there could be epigenetic factors that can play in compared to the environment, but I'm not sure of how that difference in regions can cause it. But
okay, you showed also in your presentation a number of possibilities to reinforce existing medical therapy by those cap peptides.
Yes. Yeah.
Okay, and if I think of future perspectives aimed at personalized medicine, you know, you want the optimal treatment for your tumor, not for any tumor, but for your tumor. Is it relevant to have the omics data of that tumor specifically? You know what I mean? Are those cat peptides, do they work in any stage or in any retinoblastoma tumor? Is it like a generic medication that you would prescribe? Not yourself, but...
Yeah, the caps, out of 8 caps we tested, only 2 work for retinoblastoma.
Mm-hmm.
So caps work by, these are positively charged peptides, which are 12 amino acid base in structure, and they have lysine and arginine on their outer surface and hydrophobic residues. So they target negatively charged cells or negatively charged phospholipids on the cell surface, which is the phosphatidylserine. So these positively charged peptides go and bind to the negatively charged phosphatidylserine expressed on the cancer cells. Normal cells do not express phosphatidylserine on their surface, but cancer cells expresses because they undergo chemotherapy treatments or some other DNA damages. So these are expressed on the surface. So that's why it can be used to selectively target cancer cells and spare normal cells.
Okay, and I think you don't want to interfere with the, for instance, the blood-retina barrier. But could you inject medication into the eye instead of giving it systematically? No.
Yeah.
Would it be too dangerous? You think?
We haven't tested it in the animal models to confirm it, but these should be able to cross the blood-retinal barrier, because there's a recent study in last year in I think from the University of Shanghai. They have used similar peptides, which is called a penetratin, and they caused tryptophan and some other amino acid mutation. And they used it as a topical installation with melphalan to target retinoblastoma. So that uses a non-corneal pathway to infuse into the eye and reaches the vitreous.
Okay.
And they showed a 24-hour absorption rate in the vitreous. So it's possible to use, once we modify the caps, we can still use it as a topical installation.
Okay. So, and if you can't get hold of any material of the eye, of course, because you don't get a biopsy or whatever. Is there any link between your findings and, for instance, known systemic biomarkers in retinoblastoma? You know what I mean? If in blood or plasma, there are biomarkers for retinoblastoma. Yes. Is it linked to your findings in any way?
No, not much. So the follow-up study for my findings will be to develop tear-based biomarkers for retinoblastoma.
Okay.
Yep.
And is there any link between clinical data and your findings? Is our OCT helpful in the earlier stages?
Yeah, fundoscopy and OCT is helpful at early stages, but the key findings of our experiment shows these tumors does not follow a glycolysis metabolism. This is why retinoblastoma tumors are not subjected for FDG PET scans. because they do not take up the FDG dye since they have low hexokinase system. So that's one peculiar nature of these tumors where we cannot use PET scans for diagnosis.
Okay. Well, I give the word back to the pro-rector. Thank you.
Thank you.
Thank you very much, Professor Webers. The opposition will be continued by Professor Mohan, who was also a member of the assessment committee. He's professor of ophthalmology and molecular medicine at the University of Missouri in Columbia, United States. Welcome, Professor Mohan. The floor is yours.
Thank you very much. And I want to congratulate Dr. Babu for a wonderful presentation and also a very exciting work as well. So you have made all this presentation. I just have a few questions, like what stages of the RB patient included in the study, which you already explained it, D and beyond. So do you see any similarities between D and E, or you just took both D or beyond, like terminal cases?
We don't see much similarity in their expression profiles because we tend to find around 1,800 genes that are unique only, significantly deregulated, and are unique only for advanced stages, which is the group EIs, compared to the group T. So we have different expression profiles for group E and group T tumors, where they still show a difference. Like, for example, the microRNA 181A5P, which I showed, they are significantly downregulated in advanced tumors compared to the group D tumors.
Great, thank you. So the other question, which is kind of related with this, so you do see the molecular changes on these in your samples. Do you have any prediction or idea? Do your results suggest that when these changes start during development of RB tumors?
The changes mostly, development of tumors should start with the mutation of the RB1 gene, which should be followed by the dysregulation of various genes, which involves hyperactivation of oncogenes, followed by chromosome instability in the photoreceptor layers. I believe that's the initiation point of the tumors. You need to have RB, loss of function of RB, followed by hyperactivation of oncogenes to further support the tumor initiation.
Yes, but can you predict the timeline for the patient? That's based on your results. Do your results allow you to predict any timeline that this is what it is, and in future, you have 5 years, 6 years, 10 years to go to these different stages of the RB?
We are currently developing a biomarker which can predict the severity from the tears, but that has not been added to the thesis study yet. But yes, you can do that with tear-based biomarkers, where we can predict the severity and the therapeutic effects on the retinoblastoma tumors.
I really like your metabolic reprogramming, what you have done in the experiment, but I'm not expert in it. So would you mind giving a little bit more details, like how it can be used more in the relevance of RB?
So what we find in the microarray and metabolomics integration analysis is RB tumors are more inclined to fatty acid metabolism. This is specific for the advanced tumors. When we expressed RB back in the cell lines, we see an induction of hexokinase, which is one of the key enzymes in glycolysis. So we further looked at their metabolic phenotype in RB null and RB overexpressed conditions, and we find that in the RB overexpressed conditions, these cells induces hexokinase and further elevates glycolysis, At the same time, they activates AMPK and shut down fatty acid metabolism. So this reduces the amount of ATP turnover from the fatty acid metabolism for these cancers to glycolysis-derived ATPs. So by using a metabolic flux analysis and signaling pathways, we see that switching the primary metabolic pathway, which is the fatty acid metabolism, to glycolysis causes severe effects on these retinoblastoma cells, where it functionally affects its cancer hallmark property like proliferation, invasion, migration, and they become more sensitive to chemotherapeutic drugs. So we see that reprogramming the metabolism can further use for other hallmarks of cancers. Like, for example, if they use more of glycolysis, as we said, when we reprogram it to more of glycolysis, we can further look in the future if how they behave to the immune cells in the tumor microenvironment. If they are more inclined to fatty acid metabolism, is the mechanism they uses to evade infiltrating immune cells to their tumor microenvironment. So these are futuristic things we can do with the metabolic reprogramming in cancers, where it's always interconnected to the other hallmarks of the cancer.
Wonderful. So in an essence, what I'm getting it, like you were able to identify a molecular target that can be used for new therapy development for RB.
Pardon? Pardon, can you repeat the question?
Yes. So in an essence, what I'm understanding that you were able to identify the targets that can be used for new therapy development for the RB.
Yes, like I said, one of the key findings is that reprogramming metabolism can help you make these tumors more efficient or more targetable to conventional chemotherapeutic drugs. But I would still suggest to do more in deeper studies to further understand their tumor microenvironment, to further go into therapeutic strategies.
Great. So the other question is, what are the benefits of using AICAR treatment over the metformin? Metformin is already used for the diabetes and all that, for hyperglycemia. So why you pick this AICAR?
So we did the experiments with metformin as an AMPK activator. So pharmacologically, we can activate AMPK using both metformin and ICAR, but ICAR is a more direct activator of AMPK than metformin. Comparingly, metformin, when we did the in vitro assays, we find ICAR is more effective in reducing tumor burden in vitro, in the in vitro analysis compared to metformin.
Nice. Nice. That's a good strategy. I mean, this is nice. And what do you think, like, a future of treatment for RB goes from, how it will change the field for the treatment, from bench to bedside translation, how useful your study would be?
Yes, for the diagnostic purpose, like I said, these don't follow an FDG PET scan pathways. But what we find is they have more of fatty acid metabolism driving tumors. We can use one of the metabolites of fatty acids as a diagnostic marker to, like a PET scan, to do the diagnosis of retinoblastoma markers like an L-carnitin DG scan or something. That's one possibility. The treatment point of view, we still need to work on the fatty acid oxidation pathways. The current thesis, we worked only on the fatty acid synthesis pathways, but then there is fatty acid oxidation pathways. If we can target that and further add more chemotherapeutic or low dose chemotherapeutic to target the drugs, or a treatment strategy where we can completely avoid chemotherapy and use only drugs that can selectively target retinoblastoma tumors, are going to be the futuristic for retinoblastoma research.
Thank you. So the last question I have, which is not actually this is just a very out of context question, I would say, a more ambitious question. So do you think, all these AICR treatment, all these different treatment which you find useful are blocking this pathway, can be used as a prophylactic when patient is at the stage B or C? And it's just a very ridiculous. It has nothing to do with your thesis or anything. It's just, I'm challenging your thinking, like what you see where the future can be.
I understand your question, but I wouldn't recommend using ICAR at an early stage unless we know that group A or B or C are following a fatty acid metabolic pathway. We still don't have an information on that stages of the cancers. All our study was focused on group D and group E. If the initial tumor formation follows a metabolic pathway like a fatty acid dependent one, then ICAR would be an effective treatment strategy where you can activate AMPK to block fatty acid pathways. But here it's again tricky because AMPK is a metabolic sensor. It can work to activate the pathway and it can also work to inhibit the pathway depending upon the metabolic landscape the cell requires at that stage at that time. So I wouldn't recommend using ICAR for early stages.
Very smart answer. I really loved it. Thank you. This is the right answer, actually, what you just said.
Thank you, Professor Mohan, for your contribution. The opposition will be continued by Dr. Gijs. She's assistant professor at the Department of Ophthalmology at this university.
Thank you. Dear candidate, I would like also to want to start with complimenting you on your nice piece of work. Especially, I like that you use different technologies and that you also use different techniques to validate your findings, which make that your findings are very strong and solid. And also compliments to the graphical part of your thesis, especially the graphical abstracts. They are really helpful in understanding all the pathways. It's a very nice, colorful work that you made, and compliments for that.
Thank you.
I do have a few questions. The first one is it's on chapter 2 about the transcriptomics and metabolomics study.
Mm-hmm.
Why did you decide to do metabolomics instead of proteomics?
Thank you, esteemed opponent. Thank you for your kind words and question. We tend to look at metabolomics rather than proteomics because we wanted to understand the current changes in the tumor as a part of the gene expression. So the understanding metabolomics would help us to identify small molecule therapeutic targets for the tumor, and gene expression will provide the backup information of the gene regulations. And using multi-omics, we can still integrate the gene regulation and metabolic or metabolite regulations in the tumor. So we tend to use metabolomics than proteomics, but we have plans to use targeted proteomics in the future, in the coming days.
Okay.
Yeah.
And if you would repeat the transcriptomics study right now, would you go for RNA sequencing instead of the microarrays, which have become already a bit outdated today?
Yeah, we'll go for a single-cell RNA-seq probably because to understand more deeper on different cell types in the tumor. That gives us more idea of how the different types of cells rather than tumor cells or different types of infiltrating immune cells. So we can categorize them using a single-cell RNA-seq.
Yes. And the results of that study was that the glycolysis was low and the pyruvate levels were also low, which was a bit surprising to me because you would think that cancer cells need a lot of energy, so the first source that they would go to are the sugars, but apparently they are not. Is this because of tumor necrosis in the center, because the tumors become too grow that they don't have oxygen supply in the center?
Well, But when it comes to cancer metabolism, globally, what we see is these cancers tend to deviate from the metabolism of their parental lines. So, like for example, when lung cancers originate, they tend to deviate from the metabolic pathways of airway epithelial cells. Similarly, these tumors arises from retina or the photoreceptor layers of the retina. So retina is the highest glycolytic metabolic tissue in the body next to brain. So they utilizes more of glucose for their rapid changes and cellular biochemical pathways and even the vision cycles. So a cancer arising from retina deviates from glycolysis and uses a different metabolic pathway.
Okay, I see. Then I have some questions on chapter 3, the xenografts model that you made in the rabbits.
Yeah.
When I was doing xenografting in mice during my PhD, I was always told to use matrigel when injecting the cells in order to prevent them from spreading around and getting metastasis. I did not see that you use matrigel.
Yeah.
Did you?
No, we did not use matrigel. This was done in Singapore Eye Research Institute. We directly injected them into the intravenous or into the vitreous chamber, and we used rabbit eyes rather than mice.
Mm-hmm.
So these cells are more suspended, and they tend to go and sit to the retina once you inject. And over the time, across 6 to 8 weeks, they tend to grow from the retina. And that's how they
Yeah,
use it.
and I see that you measured the growth over time, so the tumor growth was monitored, and it was expressed in your thesis in pixels
Yes.
rather than diameters.
Yeah.
How was it measured, the tumor growth, and how did you get an idea about the depth of the tumor?
Yeah, so we used OCT and a RETCam to get the images, and most of the tumors that grow, they are not they are multifocal. They grow in multiple colonies in the retina. So that's why we measured it in pixels rather than in diameters, because it's not a single tumor growing there. There are multiple tumors which are in different sizes, but they grow across the region of the retina.
And how do you get an idea about the depth or the?
I'm not sure of the depth. Yeah.
Okay. And did you know that the rabbits were also monitored for their visual functions and, for example, the IOP, intraocular pressure?
Yes. We measured the intraocular pressures, but they normally didn't show much. They don't form much larger tumors because we didn't expect them to grow large tumors. We just wanted to see if the cell lines are able to grow tumors. And to an extent, for the therapeutic models, we grew tumors and then immediately treat them. So 6 to 8 weeks we give them for the tumors to grow, and another 2 weeks of treatment. So we didn't measure much of intraocular pressure at that point.
Okay, I see. Then I also like chapter 6 about the peptides, where you developed or designed firstly 2 new peptides and then tested them in vitro. So they do not cause bystander toxicity to the retinal cells. But what is then their disadvantage of using them? Yes,
so these peptides are told amino acid base pair peptides, which forms an amphiphatic structures, and they also have lysine and arginine on their outer surface, and they have hydrophobic residues. So the advantage of these peptides is they are positively charged. So they target a negatively charged cells or a phospholipid on the surface of the cell, and using these hydrophobic chains, they can integrate into the inner membrane of the cell lines and selectively targets cancer cells. So what we see is they are able to induce apoptosis in retinoblastoma cells and spare normal cells, because retinal pigment epithelial cells, what we used in this study, they don't express the phosphatidylserine or the negative phospholipid on their surface, but retinoblastoma cells do. So that's how this mechanism works to selectively target the cancer cells and spare normal cells.
Okay, so it's about high selectivity and specificity.
Yes.
Okay. And how do they get into the nucleus in order to cause DNA damage?
Yeah, so these hydrophobic chains help them to go inside the cells, and what we see is they co-localize mostly in the mitochondria rather than in the nucleus. So since the highly active organ in the cell is mitochondria, they're more of negatively charged, and these peptides go and bind to the mitochondria, and further it causes a caspase-mediated cell death in retinoblastoma cells, but we don't see them co-localizing in the mitochondria of retinal pigment cells. So their localization differs, and they also selectively targets retinoblastoma cells.
But the DNA damage is the nuclear DNA or the mitochondrial DNA?
It's a nuclear DNA damage. So they once the caspase-mediated cell death and the nuclear DNA damage is all.
I see. And you investigated the synergistic effect of the peptides together with the chemotherapeutics, and therefore you get like an FICI index.
Yes.
Which can be either synergistic or additive.
Yep.
If it's additive, would you then recommend it as a therapy, as a combination therapy, or does it really need to be synergistic in order to translate it into the clinic?
We still use it even if it's additive. If it's synergistic, it's very good. If it's additive, then we need to still add a much more higher concentration of the peptides to induce the effect of the chemotherapeutic drug.
Yep. Okay. And then I also have a question about the blood-retinal barrier. Do you think that they would disrupt the blood-retinal barrier if you would inject them systemically? Because of their charge?
We still don't know, but few other studies done in a different lab, they have shown that they were able to, topical installation of charged peptides can infuse into the retina using a non-corneal pathway to reach the vitreous chamber. So we haven't done it, but I think if that's possible, then we can still modify our peptides and try it out.
Because is there anything known about disruption of the blood-retinal barrier in retinoblastoma patients and metastasis?
Not much. The advanced tumors do are more invasive into the choroidal and the optic nerve, and it also touches the lens. So it's all full in the globe, and then it further metastasize to the distant organs. So it still disrupts, and they also form vitreous seeds, small tumor cells in the vitreous.
Yep.
Yeah.
And then, do I have time for one final question? One more, okay. Well, I want to ask you if you know about the lens cells. They don't develop spontaneous tumors. What can you learn from those cells in the context of the retinoblastoma tumor formation? Would the comparative study be shed new light on it, or?
You mean if we can model an initiation of retinoblastoma tumors?
If you would do, for example, a transcriptomics study with lens cells and retinoblastoma tumor cells, what the differences would be there, and maybe if the retinoblastoma protein, if it has a role in the lens. Any thoughts on that? Yeah,
it's possible, but I don't know if we haven't looked into that angle.
No worries.
Yeah.
Okay, thank you. I will give the word back to Pro-Rector.
Thank you very much, Dr. Gijs. Then it's time for the last opponent, Dr. Nabbe. She's assistant professor at the Department of Genetics and Cell Biology at this university.
Thank you very much. Well, first of all, I would also like to congratulate you and your team of promoters, but also your family, who can be very proud, I think, at both your defense but also of this all the work that is in this book, of course. I think it ended up in being a very impressive thesis. It's already mentioned by the other opponents as well. It includes a lot of work coming from patients, animals, cells, going to treatments all the way, and and with all types of techniques as well. So that's very, very nice. And I think it resulted already in 5 publications, of which 4 you are the first author, what I understood.
4 publications. Yeah. 5th one is still in the press. Yeah.
There you go. So I think it's really nice. That shows that it's good work what you have in here. But I do still have some questions, and they are mainly related to chapter 3, which is one that is, well, maybe now being published, but not yet, at least when I saw the book. Yeah. My first one is, I don't have a background, I don't know anything about the eye. I have a background in cardiac metabolism. Mm-hmm. And I was a bit intrigued by, you also mentioned it a couple of times, but also figure 7 has it in the title. It says AMPK alpha activation inhibits fatty acid metabolism. Yeah. Do you have any idea why I'm surprised by that title? Or?
Yes.
Yeah? Can you explain or speculate a bit on that? Or
yes. Yeah. So AMPK activation, like I said before, it differs with the cell type. So here we see the activation inhibits fatty acid metabolism. This is not the same even if you look at the glioblastomas. Maybe AMPK activation maybe favors tumor growth. So it differs within the cell lines, and it's differ. And what we find it's quite unique in retinoblastoma tumors.
And how does it work then? Like the pathway-wise, like in the heart, for example, you have the AMPK effect on CPT1, on the CPTs. How is that? So AMPK1
inhibits acetyl carboxylase CoA, which is the ACC gene. So ACC gene is a fundamental gene which is required for fatty acid synthesis. So inhibiting fatty acid synthesis has effect on malonyl CoA, which further gets converted to for the oxidation pathways of fatty acids. So the oxidation of fatty acid further provides acetyl-CoA into the TCA to run the mitochondrial engine. So if the cell is dependent on fatty acid metabolism, their main pathways that provides acetyl-CoA to run mitochondrial TCA will be fatty acid synthesis and fatty acid oxidation. So initial hit is activating AMPK to inhibit ACC. So the synthesis stops, then you don't have much precursors to run the oxidation cycle. Yeah.
Okay, that is clear. And then my second question is also a bit related to that, because you talk about this shift in metabolism there between fatty acids and glycolysis, basically. But I was wondering, in relation to your chapter 3, what are the experiments then that show that there's a shift in metabolism?
Yes. So in the chapter 3, we did biochemical assays using Western blot to show that the signaling mechanisms are different upon RB overexpression and in RB null cells. And to functionally validate it, we used the metabolic flux analysis, where we measured oxygen consumption rate and extracellular acidification rate separately in hexokinase overexpressed cells and RB overexpressed cells and hexokinase null and RB null cells. So the oxygen consumption rate was higher in RB null cells, which was lower in hexokinase overexpressed conditions. And similarly, ECAR was higher in hexokinase overexpressed cells, and RB null cells has low ECAR. So oxygen consumption rate represents the mitochondrial OX-force, and ECAR represents the glycolysis metabolism. So that gives us an insight that the differences or the shift happens when you overexpress RB or hexokinase.
Okay, but then in relation to that, because it's indeed the COs what you used, right? And the mito stress test is next to the ECAR?
We did a mito stress test, we did a glycolytic rate assay, we also did a mito fuel flexibility assays, and we did 3 different COs assays to confirm the fuel usage and the metabolic reprogramming part. Further, we also measured the metabolites produced by these cells using ELISA. So these RB overexpressed and hexokinase overexpressed produces more of pyruvate, but the other ones have low pyruvate. And the RB null cells have more of L-carnitin, which is a key metabolite of fatty acid metabolism. RB overexpressed and hexokinase have low L-carnitin. So similarly, we measured few other metabolites, which further confirms that there is a shift in the metabolic pathways.
Okay, I guess you partly answered my question already, because my question was also in relation to the seahorse assay.
Okay.
What is in that medium that is in there? Like, what are you measuring that is being oxidized?
Yes, so we use a plain medium without any glucose or a glutamine supplement, and then we subject them for the seahorse assays. In the medium, we don't measure anything, but we using the kits, we measure the oxygen consumption rate and the mitostress, the glycolytic rate assays, and the mitofuel flex assays are provided with inhibitors for 3 different the metabolic pathways. So that helps us to measure which pathway the cell line is dependent and flexible to use which fuel to run their TCA cycle.
But I mean, there should be something in your medium that is being oxidized, right?
Yes.
It's not the fatty acids then?
It's not a fatty acid, yeah.
Okay. My third question, I have to search for it. It's actually my fourth, because I already answered a bit. In relation to the treatment that you were planning on doing with the AMPK activators,
yes.
How do you envision that? You already said that you have to be a bit careful with giving those, of giving ICAR, I guess. Or what would be your ideal treatment if you want to go targeting metabolism?
Yes, like I said, giving ICAR at the early stages, it's still not recommended because we don't know how the early stage tumor metabolic landscape is, because we haven't discovered it. What the thesis study, we use group D and group E eyes, which are already at an advanced stage. So those, we tend to find that they have a low glycolysis and high fatty acid metabolism. So we can use ICAR to activate AMPK, which further inhibits fatty acid metabolism. And if you use it in combination with chemotherapy, so the reduction of inhibition of fatty acid metabolism reduces the total ATP turnover of the cells, mainly drains its energy. And when you give low-dose chemotherapeutic drugs in combination at the same time, that is a more convenient and effective treatment strategy to manage tumors then.
Okay. And how are you planning on, and that was something that you just mentioned or earlier, about as a diagnostic marker, you want to focus on metabolites of fatty acid oxidation, you mentioned.
Yes.
What metabolites were you thinking of measuring?
I would go with L-carnitin because we see that as a top hit and one of the key metabolites which we find in our in vitro assays. Like they use FDG glucose, if we can develop a kit that revolves around a fluorescent L-carnitin, which will be beneficial to diagnose these retinoblastoma tumors.
Would it be beneficial for other type of diseases as well? If you think
if they use fatty acid metabolism, yes, that would be beneficial.
Okay, so maybe for the heart then,
yeah.
Would it be possible? I don't know. I still have a bit of time, I see, so I still have a couple of questions that I would like to ask then. If you get a bag of money, what would be your follow-up experiments? Let's say you go for an NIH grant, which is pretty big, I think. You're now in the US, right?
Yeah.
What would be your first experiment to follow up?
I would develop a line, so the main key goal is to identify the early stage retinoblastoma tumors. So right now I know the systems to develop early stage tumor systems. So I would rather do iPSC based retinal pigment epithelial lines or photoreceptor. I'll develop a photoreceptor line and knock out RB tumor suppressor gene to initiate retinoblastoma tumors, and further overexpress oncogenes to induce tumor initiation, and then look at their metabolism, because the major difficulty we have is we don't have a photoreceptor line to look at and to study the initiation of retinoblastoma tumors. It's challenging. You need an iPSC based system to develop photoreceptor lines and further knock out RB to see if they can form retinoblastoma tumors.
Thank you, Dr. Naber. Vishnu Babu, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return in this room.
Okay, thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Vishnu Babu. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Heijmans is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
So by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Vishnu Suresh Babu, the degree and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Please be seated.
So before I leave the word to Dr. Ghosh, your main supervisor, I would also like you, Vishnu, to congratulate you, so I should say now Dr. Suresh Babu, to congratulate with your fantastic thesis. When I learned to know you in Bangalore as a very kind and gentle and modest person, I was really rapidly impressed by the amount of work you had done and also the way you presented your work, and as we discussed in the committee, the way you defend it with very precise, in-depth answer, very honest also about your knowledge when you know something and also when you don't know something. So I would also like to congratulate, of course, your family, because all the work, your life is all, and also your personality is not only genetically but also environmentally determined by your family. They really have to and should be proud on all the work you have done and your performance today. With that, I would like also to leave the word to Dr. Ghosh.
Thank you, Dr. Heijmans, and thank you to the university and the committee for this wonderful gathering and the evaluation of this work. So I'm really proud of Vishnu, Dr. Vishnu Babu, that is now completed this journey, and it really warms my heart to reminisce some of the days of our journey. So Vishnu was one of our first students, and he was the quiet one. So initially I would be quite concerned that this boy doesn't speak much. So is he enjoying the work or not? So, but over time, he really took ownership of the retinoblastoma project. I'm really proud to say that he himself has made quite a few seminal discoveries, and I remember our discussions together where he would do a lot of reading on his own and then bring some ideas to discuss with us, and that's truly a mark of potential for future scientific independence. So I think that is where Vishnu is headed. I hope he continues to grow. As I was telling the committee also, I was surprised by a little bit surprised. He always had the knowledge, but I was surprised that he spoke a fair bit for some of the answers he gave. Usually he is much more shorter in his answers. So Vishnu, apart from this, he was a fantastic, part of our lab. He used to be in all of our meetings together, in all of our celebrations together, and would bring food. And I know his mother is there. And we from the lab used to look forward to his mother because she's a wonderful cook. And she used to feed us every time she would come to feed Vishnu. So we were spoiled by her. So in all, I think Vishnu has achieved what we had envisioned for him, that to make a key contribution to a neglected or a rarely researched topic like retinoblastoma. And I really hope that some of this work that he has done will lead to some clinical translation. So with that, once again, congratulations, Vishnu, and congratulations to your entire family. This is wonderful. This is, I am so proud of Vishnu right now. And thank you so much, Stephen. Really, your help and guidance has helped this journey immensely.
Okay, thank you, Dr. Ghosh. Dear Dr. Babu, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. I understand you haven't been much in Maastricht at all, so maybe it's even your first visit.
Yep.
Is it? Oh, I hope you like the city, and maybe you come back someday. Anyway, I hope you have good memories of this ceremony. Yep. You didn't look too nervous, so probably you were okay. Of course, I'd also like to congratulate the family. I also hope you enjoyed the ceremony and are proud of this achievement, because it's an important achievement for someone who works in science. I wish you a lot of success in your future career. First of all, with a postdoc in the United States, yes, I understand?
Yeah. I'm a postdoc at the University of California, San Francisco.
Okay. Well, we look forward to seeing more of you, publications anyway. I'd like to thank all the members of the committee here present. Thank you very much. Congratulations to the supervising team, Dr. Ghosh, Professor Heijmans. Dr. Mohan, thank you for being present online. It was great to have you. And with this, I'd like to close this academic ceremony. Okay. We will. Dr. Ghosh and Professor Mohan, if you still want to congratulate the candidate, you can.